Alright, so we're looking at the notes today of Biology B2B. Take a minute and set up your notes page. Pause this if you need to. So B2B says, and it's a really long teak, B2B, and I'm going to edit this quite a bit so it'll be something reasonable that we can write down. B2B from the state of Texas says that you need to know hypotheses are testable. Hypotheses are testable statements that must be able to be supported by evidence. And when I say evidence, I'm also talking about observations, things that, you may not be able to go online and look it up, but you can see that you've done some things and you've observed things in nature that you can have. So it's got to be supported by the evidence that's here. And it must be repeatable. So a hypothesis has to be testable, it's got to be supported by evidence, and it has to be repeatable. The hypotheses, if you have some hypotheses, they don't grow up to become theories. Hypotheses that have been thoroughly tested can be incorporated into theories. So if they're really good hypotheses, they prove to be true over and over and over again, then they can be incorporated into theories. They become a part of a theory. Okay. So in this instance, science becomes a process. Science is actually a process, and we have a name for that process. That is the scientific method. So what we're going to do is we're going to go through that scientific method, and we're going to focus on the hypotheses that we're dealing with. So scientific method, you guys are all familiar with this. This shouldn't be anything brand new to you. So I'm going to go down through here, and I'm going to list those steps of the scientific method. So the first step, we question. We come up with a question, and I'm going to leave some space because we're going to go back and write some stuff about this, so you need to do so also. We do some research. Sometimes we can research online. Sometimes our research is observational evidence. We form a hypothesis. And then I'm going to skip a little bit more space here because we're going to write a little bit more on that, forming the hypothesis. And then we test that hypothesis. Test the hypothesis. That's our experiment. Next in line, I'm going to leave some space here. We're going to record the data. Record, and we're going to analyze it too. And after we do that, we're going to form a conclusion. Now I'm going to add one more step to this, and I'm going to put in parentheses because here we repeat. So it's not good enough to do this one time. You repeat this. In real life, scientists, they don't always follow this process, but as beginners, we go through and we make sure we follow this process. If you become a research scientist, you're going to find in reality that all sorts of weird things happen. Sometimes you get partway through and have to scrap it. Sometimes things happen accidentally, and then you have a whole new line of study to go through. So it just depends on what it is that you're doing. So we're going to start out with our question. And when you start out with this question, it needs to be very specific. It's a small question. It doesn't mean it's not important. It's just very specific. A bad question, a poor question, would be something like, how do I cure cancer? Well, geez, there are a lot of types of cancer. And so that's a huge question. It's made of a few words, but it's a big, big question because there's, all types of cancer that respond to all different types of treatment. So there's not just one simple answer. So the question needs to be very, very specific. The second part is do your research. You're going to do this research, you're going to look up and find out as much information as you can about your question. The third step is form a hypothesis. And you guys all know that the standard definition, kind of a layman's term definition, is an educated guess. This is not an opinion. You can have an opinion today and change it tomorrow just because, oh yeah, it's a different day. An educated guess, this is the word that's important here. You educated yourself right here. You did research, you made observations. It's an educated guess. This is a possible answer to the question that's testable. So it's a possible answer to your question, and it's got to be testable. If you can't test your hypothesis, then it's a crappy hypothesis. The next step on here is the experiment. And so when we say test the hypothesis, this is your experiment. It's not just a random experiment. This is looking at your hypothesis and testing this hypothesis. Am I right when I say this? And there are several parts to an experiment. We have the independent variable. And the independent variable is what you're changing. So when you did that exit ticket with the sunscreen, in the independent variable in this case would have been the type of sunscreen. Is it going to be spray-on sunscreen or the lotion? And if you don't know what I'm talking about, it means you didn't do your work. Go back in there into the remote classroom and look and see what you're supposed to have done. If we have the next one, we have the dependent variable. And I'm going to abbreviate variable. The dependent variable is what you measure. So if we go back to our experiment about the sunscreen, then what would we measure? Well, they used spray-on sunscreen or lotion sunscreen, and they should be measuring, well, how much burn was there? How was the skin impacted? So measure the amount of burn on that. The next thing would be the controlled variables. There could be one, there could be multiples, but the controlled variables are things that you have to control so they don't mess up your experiment. So for example, if you set up your experiment where you put spray-on lotion or spray-on sunscreen on one arm and lotion on the other, you would need a third arm so that you could leave it without anything on it. I'm sorry, I jumped ahead of myself. You would need to make sure that you put the same amount of stuff on each one, so the amount of lotion or the amount of spray on. You would need to make sure that you went out at the same time of the day, that both your arms when you were out there, they were in the same amount of sun. So there are different variables that you need to control to make sure they don't mess up your experiment. And then the last one is the control. And I'm going to put it in all capital letters and underline it. The control. The control is a totally separate experimental setup that you put up and you do nothing to it. It's the normal. So the control is the normal. It's untested. So if we were doing spray on one arm and lotion on the other, we would need a third arm to have nothing on it. And that would be your control. This is what it looks like when nothing is done to it. So we have the control. I wish they had called these different things because a lot of people get these confused, but this is just the standard way that things are. The next section here is record and analyze your data. This is where you always, always, always write your observations. Anything that happens, whether you think it pertains or not, you're going to write down your observations. Usually you're going to put all your data in a table of some kind or a graph. You don't want your stuff just listed. You don't want it in a paragraph form because it makes it more difficult to find. You want nice, neat tables and graphs so it's easy to look at all your data and find out where it is. The next one is form a conclusion, and you do three things here. First and foremost, you're going to answer your original question. So from that sheet, page 27 in your packet that you did, it was experimental design, exit ticket. It says ET, experimental design. The first thing you need to do is answer the question. Their question was, which sunscreen worked best, the spray or the lotion? So we have to answer the question, well, which one was it? Was it the spray or was it the lotion? The second thing you want to do is state whether your hypothesis was supported or not. Because you made a hypothesis. Was it supported? You can say yes, it was supported, or no, it wasn't supported. It's not wrong for, you don't get points off because you say, hey, the hypothesis was not supported. That's not what you get points off for. And then the third piece here is you look for error. Not error like, oh, my lab partner dropped something, but error in the procedure. Where in the procedure does it need to be changed so we get better data? So, for example, when I said you needed a third arm for that, well, obviously we're going to have some issues with finding a third arm. So maybe instead you could say 3 patches on one arm. Here's where we have it with spray sunscreen. Here's where we have it with lotion, and here's where we have it with nothing. So you could have all 3 on one arm and you would deal with that. So that's a procedural error that we could do. And then the last thing on here is repeat. Always repeat multiple times. If you can't repeat your experiment and get similar results, then something's wrong. And you need to either redo the procedure or come up with a different way of looking at it. Maybe come up with a whole new entire procedure. So when we talk about these, you have to know that hypotheses are testable and that they are supported. They have to be capable of being supported by the evidence. They have to be repeatable, and that if you have hypotheses that have been proven to be true over and over and over again, we can take those and incorporate those into theories.